Happy New Year. So it's exciting to see actually so many of you here for our first seminar of the quarter. So it's my great pleasure to introduce you to our speaker today, Professor Scott Emerson. So Scott Emerson, who is an MD and PhD, is a Professor Emeritus of Biostatistics in our department. After receiving an undergraduate degree in physics and an MD, both from University of Virginia, Dr. Emerson earned a master's degree in computer science at the University of Virginia and a PhD in biostatistics from our department. He held faculty positions at the University of Florida and the University of Arizona prior to joining the faculty of our department, where, among other things, he served as graduate program director for 6 years. Dr. Emerson is active in research into methods for the design, conduct, and analysis of clinical trials. A major focus of his research has been in the use of sequential methods, both frequentist and Bayesian, and in the monitoring and reporting of clinical trial results. He served as a member of the National Academies of Science Oversight Committee for Missing Data in Clinical Trials. His collaborations in clinical research include a broad spectrum of disease areas, including cardiovascular disease, oncology, diabetes, obesity, emergency medicine, and many others. He serves on a number of government and industry-sponsored data safety monitoring boards, as well as serving on FDA advisory committees. On top of that, he's a fellow of the American Statistical Association. So Scott's talk today will be about the controversial FDA approval of the Alzheimer's disease drug Aducanumab, and he will bring a very unique perspective, and I'm sure will tell us a lot about it. Scott, thanks for agreeing, I have to say so promptly, to give this opening seminar, and we very much look forward to it.
So thanks, Erdi. He went beyond the 4 words I suggested, but that's okay. The abstract, of course, is best summed up by this executive summary, which I borrowed from the anecdote of Calvin Coolidge going to church. He was famous for not being very verbose. And somebody asked him, how was church? He said, fine. Said, what was the sermon about? He said, sin. What did the preacher say? He was against it. So that's a pretty good summary of what transpired during all of this approval. And I'm going to try to explain what that is. No question, Alzheimer's disease is a great unmet need. It's approximately 70% of dementia with a overall prevalence of about 1.6% overall. It shortens lifespan, has a huge economic impact, both in medical care and in terms of unpaid caregivers. Not much in the way of available treatments, and nothing had been improved since 2003. There have been many failed trials, 244 compounds between 2002 and 2014, including 26 that directly targeted amyloid, including 6 monoclonal antibodies. So that's the background that we have. So now, what was happening with Aducanumab? So it's useful to go back to some of the history here. So 2005 is sort of where Biogen, who is along with Eisai, owns Aducanumab. Biogen got more involved in Alzheimer's disease. 2011 had an investigational new drug application doing phase 1 trials. 2012 to 2019, they performed a phase 1b dose escalation studies where they looked at cohorts of patients, increasing the dose, changing some of the patient populations. And so an early phase trial that would be usually followed by a phase 2 study and then a phase 3 study. Instead, while that study was still in part going on, they began development of their confirmatory phase 3 clinical trials with a special protocol assessment, which is an agreement made with the FDA about what their requirements eventually would be for approval. And in 2015, again, while the phase 1b was still going on, they started their phase 3 studies, which will be studies 301 and 302. So I'll be talking about 103, which is the early phase studies, 301 and 302, which were to be the confirmatory phase 3 studies. A key issue that arises is that a couple years into the trial, they amended the protocol to change the high dose in a subset, an important subset of patients with Alzheimer's, which is the ApoE4 carriers. Those are the people at highest risk of Alzheimer's disease. And I'll talk a little bit more about what they were doing, but just to recognize this happened during the conduct of the trial. And then in December of 2018 was the data cutoff to do a pre-specified interim analysis that was just geared at futility. They were just trying to decide that if nothing was going on, they were going to stop the trial. Based on that data cutoff, the DSMB met in March of the following year. Futility was declared. The trials were stopped. And then in 2019 of June, they met with the FDA and said, essentially, maybe never mind. We've got some interesting results here, and what can we do about it? And in some highly unusual degree of collaboration, it's not unusual that the FDA works with sponsors somewhat, but a very high level of collaboration with the FDA, culminating in an advisory committee meeting in November of 2020. I was a temporary voting member. I'm not a permanent member of that advisory committee, but I was on that. And the members of that committee, the 11 members, 0 voted for approval, 10 voted against, and 1 abstained. So then what happened? Well, the fallout, did this go too far? Yes. The fallout, this is the stock price of Biogen as different things happened. So that big drop in March of 2019 is when they issued a press release saying the trials were futile and our Alzheimer's drug didn't make the grade. And then in July, when they announced that they were intending to go ahead and file for FDA approval anyway. Then around November 2020, there's a sharp spike and then another drop. And the sharp spike is the 3 days before the meeting when the briefing documents were posted, and people read the FDA's briefing document that the clinical reviewers said, we think this should be approved. Probably not as many people read the statistical reviewer that said this should not be approved. And then we had the vote and it dropped back down. So that was what happened there to everybody. We had the, this is advancing too quickly. Personally, I received an email with this digest from a financial analyst who I knew in a previous life, that he characterized the AdCom as a cast from that 70s show. The FDA spanked with the slide rule. That was a direct reference to me personally. And he just said that this suffered from the bad panel syndrome. So that the financial analysts were watching closely. Public Citizen, on the other hand, about a month later wrote and asked for an investigation of how the FDA came to collaborate so closely with Aducanumab. And this was just based on what the briefing document said. And so this was prior to making a decision. So then what happened in 2021? Well, the FDA delayed the date that they would respond to the application. That's this PDUFA date. It had been in January or February, but they delayed it. And that's where those of us on the committee started figuring that they were looking for ways to approve this anyway. June 7th, they granted accelerated approval based on an unvalidated surrogate of reduction in amyloid. So this is not the primary endpoint of cognition, how well the dementia was progressing, but instead a subclinical marker of brain amyloid. Their indication did not restrict the approval to the sorts of patients who were actually tested in the clinical trial. They left that open. In their package insert, they presented very misleading results, incomplete in what they were. They said that the confirmatory trial generally required for post-marketing after accelerated approval was not due for 9 years. Biogen announced that the cost of this drug would be $56,000 a year for the drug. Then with all the other medical care would go to an estimated to be about $100,000 a year. And several prominent members of the advisory committee resigned in protest. By late June, 2 congressional investigations were announced about why the FDA took this action in the face of the scientific community's findings. FDA eventually, under pressure or not, but they themselves asked the Health and Human Services Office of Inspector General to mount an investigation. July through now, multiple insurers, Blue Cross Blue Shield are separate agencies in different states, but North Carolina, Michigan, Rhode Island, Florida, Massachusetts have all said that they will not cover Aduhelm. Prominent hospitals, including the Cleveland Clinic and Brigham and Women's, I believe it is, and Cedars Sinai, I don't have them off the top of my head, said that they will not use it. In November, the CMS, which is the administrative arm overseeing Medicare, announced that because this cost was so high, that if they had to carry these costs, they had to raise Medicare rates by a much higher level than they tend to raise just due to inflation or whatever. November 15th, there started being, some question about what was going on at Biogen. The chief scientific officer retired. Speculation in the press was that he was sort of forced out. There have been other layoffs due to Aduhelm's not getting the penetration it was. December 16th, the European Medical Agency recommended not to approve Aducanumab because they didn't feel the evidence was there. In response to the low penetrance on this, Biogen has announced a 50% reduction in price. And we are now awaiting a decision from CMS regarding Medicare coverage. And so if we add on what happened at the time of the accelerated approval, the Biogen stock price went way up, and now it has since fallen because it's just not getting the use that they had hoped for. So what I want to do is in that background, talk about things, and truly, my major motivation is there's a lot of things that this just did not pass muster because a lot of mistakes were made in mounting the clinical trials. And if they had done a better job mounting the clinical trials, then I would have predicted that by, instead of March 2019, it would have been the end of that year perhaps, but we would have a more definitive conclusion that the drug did work or it didn't work. But there were several issues. So that's really what I'm going to be talking about. So my basic premise, statistics is about science. And so we should just be talking about what statistics do to answer an important scientific question. And of course, science is about proving things to people. And ultimately, the validity of any proof rests solely on the willingness of the audience to believe it. We are talking about drug discovery. So we're trying to identify new drugs, and therefore there's a process that we follow, a series of experiments to establish the safety, efficacy, and eventually the effectiveness, how the drug will actually work when it's out in practice. The US regulation of drugs and biologics has a 120-year history almost, starting with just labeling, and then the drugs had to be safe. But starting in 1962 with the Kefauver-Harris Amendment was the requirement that the drugs had to really be efficacious. And this wording that I've highlighted here from the Kefauver-Harris Amendment, adequate and well-controlled investigations, including clinical investigations. Adequate and well-controlled is a buzzword used by the FDA, that if they tell you your clinical trial is not adequate and well-controlled, that means they're not going to accept it as evidence for the eventual approval. And the plural there is also all important, that it's not just a single trial, but that you have to have multiple ones. Additional amendments have addressed pediatrics and risk evaluation mitigation strategies and so on. Just so you know, I'm not going to go through this. The laws about devices are exceedingly weak. And anytime you see somebody who's doing something very, very questionably statistically and then say the FDA accepted this, I promise you it was the devices group, not the drugs group, because the laws are very weak about what is required for devices. So we were talking about getting a new treatment indication, and an indication has 4 parts. There has to be a disease, and of course, how we define a disease changes over time because of different methods of diagnosis, or that we refine our diagnosis to isolate specific types. There's a population that we have to consider because of other concomitant disease that they might have, or just demographics, such as pediatrics are different from adults, and pregnant women are separated out because of that as well. Then we have a treatment or a treatment strategy, and lots of things go with the treatment. Formulation, administration, dose, frequency, duration, ancillary therapies. Sometimes a treatment is defined by how you should change your dose in reaction. Insulin, for instance, is something that we don't give a single dose of. Instead, we tell people how to titrate their dose to deal with things. And then we have an outcome. And the outcome is ideally a clinical outcome that's important to us. It measures the way you survive, function, or feel. So that clinical aspect. But there are some outcomes that we are now accepting that are really surrogates relative to the clinical outcome. High blood pressure is one of those that we're often treating a lot of asymptomatic high blood pressure, but we feel that doing so addresses the clinically important outcomes. But notice that the outcome also depends upon the time frame, and it also depends upon the method of measurement. Our ideal results, well, we want to diagnose a new drug in which there's a low probability of adopting ineffective drugs, a high probability of adopting truly effective drugs, and a high probability that adopted drugs are truly effective. So in terms of our usual statistical way of thinking of those, we're going to use both frequentist and Bayesian criteria. The sponsor wants a high probability of adopting any beneficial drug. That's high frequentist power. Assuming the drug works, we want a high probability it's accepted. Regulatory wants 2 things. They want both a low probability of adopting an ineffective drug. That corresponds to the frequentist type 1 error. And we want a high probability that adopted drugs truly work. And that's the Bayes posterior probability. Public health, we're interested in all of those, but the other idea is we would like to maximize the number of good drugs are adopted and minimize the number of ineffective drugs adopted. Those first 3 are rates. I also consider what should we do in terms of, with finite resources, doing the best in terms of number of new drugs found. So it's both frequentist and Bayesian statistics, and they're related. In a very simplistic model in which either a drug works or it doesn't, and that our decision, therefore, is it's a good drug or it's not a good drug, and that there's only one level of it working. The positive predictive value is related to the prior prevalence that you're trying something that's a good drug, and then the frequentist power and the frequentist type 1 error. And if rather than talking about the positive predictive value, the posterior probability, if I talk about the posterior odds in this very simplistic setting, it's the Bayes factor times the prior odds. And that Bayes factor is a measure of how much you've learned. And so it is the power divided by the type 1 error. The key point of this is inflation of the type 1 error has major impact on the probability that an improved drug truly works. It's multiplicative. You have to consider the relative increase in type 1 error and not the difference. So increasing your type 1 error from 0.05 to 0.06 is a 20% relative increase. And if you want to have the same good feeling about what your posterior probability is, then you also would need to increase your power by 20% relative. So say from 90% power to 108% power to make up for this inflation of the type 1 error. So while we talk about small things in terms of a type 1 error inflation, it turns out they can have big impact on our ability to say that an adopted drug truly works. Well, this comes down to statistics and art. I remember being told, I guess it was Fritz Schultz who first gave me this quote, but in statistics as in art, never fall in love with your model. And this goes along with George Box's, all models are false, some models are useful. But lately I've been reflecting on another parallel, which is in statistics as in art, value depends heavily on provenance. When you buy a picture, a lot matters about the documentation of all the owners of that picture and really making certain that it's an original and things like that. This is true in spades in statistics. Unless we understand where the data is coming from, the resulting statistical analyses are of little value. So this inflation of the type 1 error that easily comes from data dredging, data mining, selective reporting of studies, revising outcomes to reflect the most promising results. And this can be either the clinical outcome or the statistical summarization of that outcome, whether we're going to look at the mean or the median or geometric mean or the hazard or any of those. All of these can inflate the type 1 error. Revising eligibility criteria based on subgroup analyses, changing from a surrogate efficacy to effectiveness endpoints, treating the symptom, not the disease. In order to avoid inflation of type 1 error, we've instituted a lot of things in clinical trials. We require confirmatory studies using pre-specified indication and pre-specified statistical analysis. We want detailed protocols, detailed statistical analysis plan, and registration of the clinical trial on clinicaltrials.gov if you're going to go to the FDA with your data or if you're using NIH funding. All of this is to make certain that we ask the question first and use the data to provide the answer, rather than using the data to formulate questions that were interesting. This is from Declan Dugan at a meeting that we had that we were talking about, basically things. He had a very nice summary of what is the probability of phase 3 success. And here he was showing how it differs across diseases. And a lot has to do with the practice, how they do clinical trials. A lot has to do with how successful we are, how many good ideas we have, how many of the drugs are just me too. Are we testing yet another statin, or are we really looking at a new class? Notice that neurology and psychiatry are down towards the low end, and oncology is very low. And I have lots of opinions about that, but we'll leave those off. This also matters, of course, to whether it's a rare disease or a high prevalence disease. In Alzheimer's, it's certainly not a rare disease, but it's certainly a high prevalence disease. And so that is and for those of you who don't know, I'm colorblind. And so you just have to guess when I name a color, which one I mean. But the ones that look yellow is called a chronic high prevalence disease, which we would be in there. And so you look as we go from stage to stage, what's the probability of going forward? And it's just not that unusual to have phase 3 studies fail. And I can make arguments that 50% positive phase 3 trials might be a good operating point. So you should be expected for this. This is also new molecular entities versus biologics, such as we're in a monoclonal antibody with Aducanumab, or the non-new molecular entities, some other where you're testing something else. Again, what are these success rates? This is just a point. I'm not going to go into all of this detail, but this is a thing just basically that says, if I take the same resources and 10% prevalence, there's lots of things that I can do. And in my sort of ideal world, I can explore 2,000 drugs, 200 of which work, and I can identify 160 of them and 45 false positives if I don't do screening trials. But if I go through doing phase 2 studies, and only if I've got that preliminary data going forward, then we see that, for instance, that non-adaptive homogeneous effect, I'm instead finding 165 good drugs and only 5 bad drugs. So this is very key to really follow through and do your screening to cut down on the false positive rate and to maximize the number of drugs that you actually find. Now, Alzheimer's, again, I just put this here just to revisit. Again, there's a huge unmet need that's out there. They had done, basic laboratory results. They at least told us the story. I, can't document this, but they tried to take a rational approach to creating aducanumab. They tried looking at the sorts of antibodies that patients who seemed to be doing well cognition-wise had developed, and then they tried to get monoclonal antibodies that would act in that same way. And they tried to vary some things from some of the prior failed clinical trials. And it's, of course, we now needed to test whether those attempts were valid. So their phase 1b study used staggered cohorts where cohorts of patients were 4-to-1 randomization. So they had a placebo group in each cohort, and they gradually increased the dose. So these were not concurrent. They would have finished one cohort and then started the next one. They'd have a placebo group each time, and they'd gradually increase the doses. And then some of the later cohorts, they actually started titrating the dose at first and looking at some of those things. Now, this study was eventually published, and this study was presented to us on the advisory committee. Unfortunately, the analysis that they did was not entirely valid because they pooled the placebo group across all these different cohorts while they were changing eligibility criteria. So that meant they basically built in their confounding. They built in their confounding on some of these parts. But as an early phase trial that's just to get a hypothesis generation to proceed a phase 2 study, I'll say this is done lots of times, and it doesn't raise as many eyebrows as they should, but they could have done a better analysis than this. They just didn't. But this major findings informed the phase 3 studies. They would focus on a 10 milligram dose. They wanted to focus on earlier disease. And then it turns out that trying to treat amyloid in patients leads to swelling of the brain. And it's called this amyloid-related imaging abnormalities, and it can be characterized as edema or hemorrhage. But it was noted that this area was worse in that subgroup of patients with the ApoE epsilon 4 positive. So those patients who are most prone to getting Alzheimer's were also most prone to getting the worst area. This was a big safety concern. So by looking at all of this, they decided that going forward, they would put in various monitoring. They would titrate, they'd introduce the full dose of drug gradually, but they also felt that they should give a lower dose to the ApoE4 positives. So they still called one group high dose and one group low dose, but in the high dose, you receive 10 milligrams if you were not ApoE epsilon 4 positive, and you receive 6 milligrams if you were, and so on. This was later increased by amendment. As they got their results from phase 1b completed, they started feeling like they could manage this. So the phase 1b presented the problem, presented the suggested dose, and then they went ahead with their confirmatory study before they totally understood what they could manage safety-wise. So these were the phase 3 studies, studies 301 and 302. These were identically designed. The protocol read exactly the same. It was basically a coin toss whether a given center was enrolled in 301 or was enrolled in 302. There was, they thought, no difference between these two clinical trials. So they had the placebo, they had a high dose, and they had low dose, but the definition depended upon the ApoE4 status. And they started out with 450 per arm, but using a blinded adaptive process, they decided that a larger sample size of 535 in each would be there. The ARIA management was modified by 2 of the 6 protocol amendments that were put forward. They had a formal futility interim analysis planned for when they had complete data on half the population. It was based on criteria called conditional power. They tried to estimate what's the probability the trial would be statistically significant at the end. And there was, we were not given the statistical analysis plan. The results of these trials have still not been published 3 years later, which ought to set off warning signals there. But because of that, I don't really have access to the protocol or the statistical analysis plan. And different places in the document, this was described as less than 20% probability of both studies reaching statistically significant differences, or not futile unless both studies had less than 20% probability of reaching statistically significant differences. Those two do not agree. Those two do not agree. I don't know which one was the real thing. But as this trial went on, as I said, in December of 2018, they did a database lock, and by March of 2019, that's not an unusual time span between database lock and getting the DSMB thing. You'd like it to be sooner, but it's not that bad. They decided that the trials did meet the futility criteria, and they stopped the trials. Now, what's the impact of this stopping for futility? There are some that would just say, you stopped for futility. How can you possibly come back and tell us it works? And the answer to that is, we really ought to stop saying you stopped for futility. We can do a bona fide statistical analysis. We just have to take into account that it was sequential sampling. So scientifically, this does change the time frame that efficacy and safety were observed. They had wanted to have 18 months full follow-up on everybody. Instead, they had 18 months on roughly 2/3 or more of the population, and a shorter time span for the rest. But to the extent that there might be a time-varying treatment effect, delayed efficacy, or tachyphylaxis, this does have impact on the science. Statistically, it of course reduces the precision of the estimates relative to the prior plan. But after you take into account not only the data that they had available for the futility analysis, but also the partial data that they had and the additional subjects who completed the study between December and March, they probably had 75% of the maximal statistical information available for the final analysis. It changes the sampling distribution. And I'll just note that as near as I can tell, this was what we call a non-binding futility, but I'm going to belabor this just a little bit to say, well, what if they hadn't done? Again, I haven't seen the statistical analysis plan, but this does not change how to interpret a point estimate, a confidence interval, or a p-value. Still, at the end of the day, we can get this statistical inference. And this is all subject to the proviso that the relevance of the clinical endpoint over the shorter time frame is maintained. So the criteria for futility, I think this was an error that the company made. Best choose your futility rule by looking at the inference and the efficiency and the statistical power that your sequential sampling plan gives you. A good reason to stop is because you've convincingly ruled out the minimal clinically important difference, and that the use of futility rule provides appropriate trade-off of average efficiency and power within available resources. So, look at the end of your trial. What are you going to report? I tell people, as you're selecting a sequential sampling rule, look at what your inference will be if you stop. If that inference is convincing, that's a good stopping rule. If it won't be convincing, don't do it. Now, what they used was this stochastic curtailment. And I argue against stochastic curtailment because people usually don't evaluate what they're doing and they think they know what they're talking about. And so the idea is, here is a chance to say, we will estimate the probability of having statistically significant results. And if that estimate is below a 20% probability, then we'll stop the trial. That's a very typical thing people do. They use that same threshold, whether they're doing stochastic curtailment at 25% of the way through the study or 75% of the way through the study. Notice that as I just made that statement, I didn't tell you how you were going to estimate what the future data would be. Should you presume that your alternative hypothesis is true? Should you use your current best estimate? Or if you're really bound and determined to use stochastic curtailment, my personal favorite is that you use a Bayesian predictive power. So in other words, you incorporate the uncertainty you have in your estimates so far. Based on some prior, I usually use a fairly flat prior. But they didn't really evaluate this, I say, in retrospect, because they didn't think about some things, but they had stipulated that you would use the current best estimate of the treatment effect as estimated from combining the two trials. And then that's what they would use in computing their probability. And it came out that then both studies looked futile and they should stop. Now, what should you do when you have two identically designed clinical trials like this? I always tell people, while we're used to having two confirmatory clinical trials, the fact is if one pans out and the other one's close, you may well be able to do another small study to resolve the differences, or in any case, you'll have more data to examine whether there's some possibility. So I usually urge them to look at each trial separately and go forward in that idea. But as you implement these futility rules, you have to explicitly consider time-varying treatment effects, because at the end of your study, everybody's going to be followed for the right amount of time. But in the middle of the study, we've got varying follow-up on different people. Time-varying treatment effects can happen in the monitoring of our study because over calendar time, we've accrued different patients early on than later. It's not unusual that you, in some chronic diseases, early on in your trial, you have prevalent disease. You have people who've had the disease for long standing. And at the end of your trial, you're taking incident disease. You're getting people who've had it for a shorter period of time. Things like that can happen, things just in terms of different countries getting approval for it at different times. This can happen. In study time, you're worried about delayed efficacy. You're also worried about tachyphylaxis. That's the idea that a treatment's effect wears off over time. It doesn't last forever. Here, I'll just say, is the number one thing that they didn't know what they were doing. If the treatment works, Amendment 4, where you decided to up the dose given to the ApoE4 positive people, should have led to greater efficacy, in which case you have introduced a time-varying aspect to the clinical trial. You should have thought of that. And basically, I, at this point, would have abandoned the futility rule and just let safety keep going forward, if you really believe this can happen. We could handle this amendment. They did not. Then you should also explicitly consider partial follow-up. They specified in their futility rule that they were just going to use the data from patients who had completed all of their follow-up, but there was a lot of data for people who you'd followed part way. And so long as there is not a problem with time-varying treatment effects, or particularly with patient characteristics varying over calendar time, this incompleted data is legitimately regarded as missing at random, and that we easily know how to analyze that. And there we're using all of the data. So it's best to do that. That cuts down on the amount of overrun, the chance that your data at the final analysis will conflict with what you decided based on the futility decision. So at the very least, before the sponsor considered this stuff, they should have looked at this data. And then you also should specify in this that what is your eventual inference. Sequential sampling changes the sampling distribution. You have to specify how you will incorporate overrunning data, and such issues as binding versus non-binding futility rules. If, for instance, this was there. My point is, in situations where you had a fixed sample test that you specified a single sample size and you're going to collect it all, you might have a normally distributed statistic. Depending upon the exact sampling rule and the schedule of analyses, you might, under the null hypothesis, have this lovely hypodermic needle and have what one of my former graduate students characterized as the Sydney Opera House under an alternative. So they're not shape invariant distributions, but that's okay. We know what to do with statistics. The other issue that you had to plan was multiple endpoints in clinical trials. And many of you are probably not very familiar with what we do in clinical trials about multiple endpoints. Usually we require statistical significance in pre-specified statistical analysis of the primary clinical endpoint in 2 confirmatory clinical trials to get FDA approval. Then they also want to look at supporting analyses. If there's additional doses, we like to see dose response, alternative summary measures. You might look at the hazard ratio. You might look at how many people have 2-year survival. Alternative measures of the same latent clinical endpoint. If you're doing something on atherosclerotic cardiovascular disease, perhaps you have a composite endpoint of MI or of non-fatal MI, of non-fatal stroke or death, cardiovascular death. And maybe you'd also want to just look at only the death or only MIs. And then you'd also want to look at important subgroups, perhaps. In this particular case, since ApoE4 is the highest risk group, both for getting the disease and for the safety endpoints, you might want to have supporting analyses there. Then there'd be supplementary analyses on other possible benefits. Maybe we were looking for primarily cognition in Alzheimer's disease, but maybe we'd also want to look at how well the patients could actually function rather than that slight difference, perhaps. And then we do type 1 error control through a testing hierarchy. We pre-specify the order in which we'll do the tests, and that unless you meet your primary endpoint, you don't look at the first secondary. Unless you meet your first secondary, you don't look at the second secondary, and so on. And that controls the type 1 error. And this is the most popular means to being used now. Now, the testing hierarchy in 301, 302 is not what I would have recommended. And again, I haven't seen this, but the FDA statistical reviewer commented on this. They said, first, we'll look at the high dose versus placebo on the primary endpoint, which is a measure of cognition. Clinical dementia rating, sum of boxes is what that stands for. And that's as much as I know of it. They said, first we'll check the high dose, then we'll check the low dose, then we'll look at other secondary measures of cognition, like the mini mental status exam, or this cognitive function for Alzheimer's disease, or the activities of daily living in mild cognitive impairment on that. Well, what you see here is if the high dose was the one that was going to work, you're putting an endpoint in there that's going to stop you from looking at the supportive results for cognition and other things there. So this was a very poor choice that they made, but we have to go with what was pre-specified, because if you change later, you're inflating the type 1 error. So what were the study efficacy results? Well, as I said, stopping for futility does not alter 1 iota our ability to do a scientifically rigorous, statistically valid analysis on the mean progression. And what did we see in 301? The high dose group had 2% more progression than placebo. So the direction was wrong. In 302, the high dose group had 22% less progression. And then the low dose had 15% less progression, but not statistically significant. So in high dose, we had to stop at the primary endpoint. In 301, we had to stop at the primary endpoint. In 302, we got through the primary endpoint, but it didn't pass the secondary endpoint. And then what else would we have wanted to look at? Well, these were not all available in the advisory committee documents. And during the meeting, I did actually suggest that the FDA was trying to run out the clock on our ability to ask important questions. Impact of missing data. Some of the missing data that was due to the administrative termination of the study, I would be accepting was more missing at random, but the patients who dropped out or the patients who had had to stop because of safety endpoints, this ARIA, we need to know. I also would want to see clinical importance. And that would be more important to me to see the proportion of subjects who had a major improvement in progression compared to others, rather than what the average was. See more severe endpoints. The dose response is additional ideas that I would wanted to see ITT, but you do like to get an idea of what it is by administered dose. The alternative measures of cognition, you know, just how well you think, there's a zillion ways to measure it. You know, in doing simple things, we look at orientation, memory, insight, judgment, abstraction, arithmetic, attention. All of these are different aspects of cognition that the different questionnaires would have answered differently. You would have wanted to examine what is the mechanism of action. The intended was that changing the amyloid would lead to this cognitive improvement. But you also have to worry about the unintended, that you change physician behavior. I remarked during the meeting when some of the audience was thinking that I was a cruel, heartless statistician, that my mother-in-law had Alzheimer's. I watched her for 11 years get worse and worse and worse. I watched how many times her clinicians changed the various medications she was on, and sometimes she deteriorated rapidly, that if we then just stopped those centrally acting drugs, she got better. So this is something that you have to worry about. All the other drugs they're getting, have we changed something that do that? And you'd like to see consistency across all the subgroups. Well, on 301, this was the primary endpoint over time. Being higher on this graph is being worse. So this is study 301. The gray line, I think, is the placebo group. The blue line is the low dose, and I think it's a green line is the high dose group. And you see that the high dose group is always worse than the placebo and the low dose group. So we don't have dose response. We have a point estimate in the wrong direction from study 301. The minimal clinically important difference for this particular measure is believed to be about 1. But it's not clear 1 over what time period. But here we see that we had an 18-month study, and you know, the average, all groups got worse, and the difference in the final outcomes is not very much. These are the other secondary endpoints, the MMSE, the 8S-COG-13, whatever, for the groups. And you see that basically in this study, there just was not much sign of cognitive improvement at all. And these were behaving the same. Here's study 302. Study 302 that got Biogen deciding to go ahead and try to get this approved. And so in this, you see that the placebo group does worst. You see that by the 18 months, the high dose is doing best. This difference was about a difference of, as I recall, it was 0.39 after adjusting, something like that, still well under the minimal clinically important difference. But this was a nominally statistically significant result. And we see here that as they quote this high dose group had this 0.0120 p-value. And then I put a red box around all these other outcomes. Because the low dose wasn't significant, we can't really trust the p-values because we aren't controlling the type 1 error for these other endpoints. Those are the standards that we apply. Here is a, well, let me just make certain I've got this, yeah. Here's a pseudo-responder analysis. I have to call this a pseudo-responder analysis. This is just a histogram of how the high dose group and placebo group is done. This is for the combined groups. All of that missing data is not separated by whether it's missing due to the administrative censoring or missing due to study dropout. I was not impressed with some great difference in the percentage of patients who say had less than a 1 point increase in this, and it wasn't statistically significant, but I really don't know how to do the inference on this because I don't have the data and I don't understand what led to this graph. The safety results, the ARIA was higher in both studies in both dose groups by a large amount. As I recall, it was something like 3% versus 35% at high dose. This is stuff that requires you to monitor, requires you to change the drug. It requires MRIs doing this. There was a slight dose response relationship. So the low dose had a little bit less, but the biggest jump was once you were getting this. Of this ARIA, 2/3 were asymptomatic, 1/3 was radiographically severe. Other adverse events beyond this ARIA was not that much. Maybe there was a low signal of more false. Okay, so these were the results that were pre-specified, plus or minus. Now, what do you do with those two discordant studies? Under the null hypothesis, you'd look at, what's the consistency with the type 1 error? And here's where I, get to it. It's certainly unusual to have the two studies discordant. So which is the aberrant one? Is it that it was strange that we got the significant result in 302, or was it strange that we got the direction wrong in 301? There were 26 prior negative studies for agents targeting amyloid. There was a lack of dose response in 301. There certainly wasn't enough evidence to go with this. If you presume the effect of treatment, then 301 was possibly from a different population. You were unlucky. They tried to invoke that you just got unlucky because there were more patients who deteriorated quickly in 301 on the high dose arm. They had no predictors that told you who was going to be a rapid progressor. The only way they were defined was they did worse. And with a straight face, they and the clinical group of the FDA stood up there and continued to talk about, well, if we remove the people who do badly on treatment, then the treatment doesn't do so badly. That's a tautology. I was shocked that the FDA would give this any oxygen. There was the two trials started the same time and were identically designed, but as luck would have it, accrual was a little bit faster in 301 than 302, not huge amount, but they tried to say, well, the patients in 301 did not get as much benefit from the increase of dose in Amendment 4. To me, this was very much a post hoc observation because, again, they did not change their futility rule when they put in that amendment. So there's great question in my mind exactly how pre-specified this was. Also, there were more study treatment discontinuation in 301 too. They'd been on the study longer, had a bit more discontinuation. So what did they do? Well, they went to the FDA with their results. We'll come back to this idea they did this, and they said, maybe there's an alternative pathway where we could get approval, not by having two confirmatory clinical trials, but a single pivotal trial would be sufficient. That pathway does exist, and it's meant for a time that you only do one clinical trial. It's not planned that you do two and you choose the best one and then go forward with it. But the FDA's guidance is not specific on the criteria. There have been people who've looked at this, and historically, you look for a p-value less than 0.01 on the primary endpoint, and you must have strong supportive evidence. So the FDA supported exploration of treating 302 as primary, 103 as supportive, and sweeping 301 under the rug a little bit. So the statistical issues, I referred to this as the Texas sharpshooter fallacy. They, first shoot at the barn and then you draw the target. There was no reason why 302 should have been the focus of this and not 301. So I did look at and say, well, what if you had pre-specified the following? If you had pre-specified, we will try to get approval based on both trials being significant. But if that doesn't work, then we'll look to see if one of the trials is less than 0.01. This is a multiple comparison strategy that people use sometimes. Many issues with inflation of type 1 error, but I can say that the type 1 error for 302 is now 0.02 or higher. In fact, it's substantially higher, but it's certainly not the 0.01 that they kept bandying about. And again, this is an important threshold when you're trying to do something with a single pivotal trial. The post hoc nature focusing on this, again, the lack of the dose response within 301 sort of raised their question about the dosing aspect, because in 301, the low dose group got lower dose than the high dose group, but did not have as good cognitive improvement. It wasn't statistically significant in either case, but we just were not even seeing that dose response. The deletion of subjects based on rapid progressors was totally without rigor. I have, and I mentioned this during the AdCom, I have in my life testified in scientific misconduct trials where people did this sort of thing without being upfront about it. But the point is, it's not acceptable to do it, but it's only fraud if you do it and you don't tell us about it. And then study 103, because it was not analyzed by randomization, could not be considered an adequate and well-controlled study. And it also wasn't the primary endpoint to do this. Well, why do we have to focus on this? We have lots of examples of where, observational studies, which this becomes when you don't follow a pre-specified analysis plan, observational studies are counteracted by the later clinical trials. So arrhythmias, beta carotene, and hormone therapy are the ones that I usually trot out. So I have also usually found that in the FDA, when they ask the advisory committee studies, they ask, do you find this evidence compelling? And if not, what would you find? What further study would you have? So I just offered it. And I just said, Biogen got people to come up here and say that they were on the active arm, and when they stopped, they thought that they got worse. They didn't tell us what they said before they unblinded them. But I took that at face value and said, do a randomized withdrawal study. Treat these people for a while and then take the drug away. And if all of the testimony you were giving was true, then you'd pick this out. There's lots of problems with the randomized withdrawal study, and people who work in the area say, yeah, we don't know that we'd really do this. But my point is that if what they were saying was true, it ought to be identifiable. Now, when they made their decision in June of 2021, Dr. Dunn invoked accelerated approval. I skipped one by accident. They went to accelerated approval. During the advisory committee meeting, the director of neuroscience said specifically, we're not using amyloid as a surrogate for efficacy. So there is no input from the advisory committee on what they eventually did. But they had several meetings. Office of Biostatistics was unwavering against approval or accelerated approval. The clinical reviewer and the Office of Clinical Pharmacology supported it. And they eventually approved this, apparently relying on analyses done by the Office of Clinical Pharmacology. This I seem to be skipping too many when I hit the wrong button here. Just saying that, he says, he thinks that met accelerated approval because there is some uncertainty, but the outcome of amyloid, the decreasing amyloid is reasonably likely to predict clinical benefit. So truly, prior to this, there had been no evidence in support of that. There's some general comments about using accelerated approval at all. This is the idea that you approve a drug based on a surrogate, and then there is supposed to be a requirement that a later post-marketing trial prove that it really has impact on the true clinical output. But there's been a number of papers that have reviewed the history of the FDA with this accelerated approval. The idea of enforcing that an additional confirmatory trial was done was poor. There 13% of such drugs were on market for 9.5 years without a confirmatory clinical trial. The FDA itself, a group out of oncology, reported on 93 of their accelerated approvals, and they said 51 later satisfied requirements, 37 were pending, and 5 were withdrawn. And they presented this was good, but this was censored data. This was censored data from that 37 pending. So a paper the next year considered those same 93 and said what really happened. 19 of those drugs demonstrated benefit on a clinical outcome, overall survival. 19 of them, instead of doing it on a true clinical endpoint, just repeated the clinical trial on the same surrogate endpoint, and 20, you just used a different surrogate endpoint. So when the FDA reported on their own behavior, it looked good, but when somebody else goes to examine the FDA behavior, it doesn't look good. A more recent paper said, let's look what happens to those same drugs in Europe. And of those 93, 12 were not recommended, 30 weren't even reviewed. And of the 51 that are available in the UK, 86% have additional restrictions on them. So there's a lot of questions about the accelerated approval. Now, on a surrogate endpoint, what is our goal? So they wanted to use reduction in amyloid as a surrogate for improved cognition. And what do we want to do? Well, there's two questions. Are we just trying to diagnose that a drug works, or are we trying to identify an endpoint that it improving in the individual also suggests that the clinical endpoint will improve? So is the potential surrogate indicative of a patient receiving benefit? A mediator analysis, I think, is the better way to go with this, to look to see how the treatment effect behaves after you adjust for this. But is it just a biomarker of drug effect? Well, that we can do a group level correlation analyses across a population of drugs, but are we estimating a common relationship, or is the drug a random effect that some drugs they might be related, some drugs they might? And in my usual reduction to absurdity, I just point out, in cancer, we can demonstrate that bone marrow suppression is correlated with clinical response across the drugs we approve. Although the patients with the worst suppression may get no benefit because they can't take the drug. So again, are we trying to use a surrogate that's indicative of what happens on an individual, or are we just trying to say any drug that works on A must also do this on B? The best approach is a meta-analysis across multiple classes of drugs, and do not look at correlation. Anybody who's had a class from me knows that I regard correlation as a p-value more than as a scientific estimate. Use the slopes. And then still, you have to include all available studies. You need to be clear on what's your measure of the clinical input. We said cognition, but what's your measure of cognition? Need to be clear on the measure of the surrogate. We said removal of amyloid, but how are you determining the removal of amyloid? And then we need to really allow that different drugs, different classes of drugs may not have the same relationship in terms of giving a benefit or not a benefit. Now, the OCP analysis based on correlation of group effects, they noted that an analysis based on individual data will be attenuated, but they erroneously attributed that to confounding rather than just precision. I'll also note they tried to simulate results of a comment I made about the provenance of the data. I said, given that you're only coming here because you have one positive study, what's the probability under that, if the null is true, that the other one would look this bad? And they instead just simulated, assuming it all worked and not taking into the sampling scheme. But they looked at analyses where they only looked at aducanumab studies. They took aducanumab studies and then got rid of the one they didn't like. They took aducanumab studies and excluded the patients they didn't like. And then they also took studies plus published studies of monoclonal antibodies. Now, it's interesting that they limited themselves to published studies of these others, but they didn't limit themselves to published aducanumab studies because they still have not published this. I suspect it's because they can't get it published. I expect that the peer-reviewed journals are not accepting of what they want to do, but I have no proof. I do know that they submitted it to JAMA and withdrew that. So they looked at this picture and they said, here we have on the y-axis, the measure of cognition, the improvement over placebo, and on the x-axis, the improvement of amyloid. So being low to the left on the x-axis and being low on the y-axis are both good. And we note that 301 is high. Interestingly, and just misleading things, their dot size is not the uncertainty in where the point is. It's quite the opposite. The size of the point is related to the amount of information they have. So we should be counting that high dose 301 as more important than these others. And of course, this is a lot of correlated data. The same placebo group is being used in a lot of this. But then they did use some other measures. And so here is another presentation of some other drugs, some other monoclonal antibodies, and what they were looking at here. And they argued that they were seeing good behavior across all of these. But again, this was not looking at how the improvement in amyloid was associated with a specific improvement and hopefully a clinically important improvement in cognition. And interestingly, in February of 2021, so before their decision, a meta-analysis was published that, if you ask me, did do it right. Eric Chetkin is the only name that I recognize on this, but did this, that they did a meta-analysis in order to be able to see how the cognitive change related to the changes in amyloid levels. Now, they did have to, they used the same measure of amyloid, that SUVR, that was the measure of amyloid, but rather than using the CDR-SB, they could get the MMSE. So that's what this is used for. But this is basically a forest plot of the effects. Something that's to the left suggests that amyloid reduction is harmful. Something that's to the right is amyloid reduction is helpful. Some of these are multiple things of the same drug, but different clinical trials. And then at the bottom, they gave a pooled estimate of all data, all antibody data, all published data, and all published antibody. And obviously, the two, the most promising antibody data is this Aducanumab, although Lilly has another drug that's not yet been published that has some suggestions. So you see that without the unpublished data, it looks harmful, but certainly there's not a suggestion of a difference. And again, on our x-axis, on the MMSE, minimal clinically important difference is maybe 1, but this meta-analysis is doing it the right way. There are still many problems. We don't know, or I don't know, the similarity of the patients in these different trials. I don't know the similarity of the time, but just averaging across all of that, there's not much evidence in support of this. So final comments. Statistics means never having to say you're certain. And in particular, it is not my burden of proof to say that I've proven that Aducanumab doesn't work. What I can say is it's proven that there is a safety signal that is important, that we would not want to give this if it didn't work well, and that the evidence they presented is not adequate. Biogen is reportedly going to start a confirmatory trial this spring. To my mind, they've wasted 3 years of valuable time as they sought to have substandard data accepted as evidence. Hopefully, they'll do a better job at planning and implementing a protocol. But Biogen is under the gun now, as I stated earlier. They've had, they've had to do layoffs and things, all because their projected sales of Aducanumab wouldn't work. But at the end of the day, I still quote my favorite statistician is, you just have to think about what you're trying to do. And I do this especially for Navneet, because he would be really disappointed if I gave a talk and didn't show this slide. So thank you.
Thank you, Scott, for the terrific talk. Time for questions. Well, we are over time, 15 minutes, but maybe a few quick questions. Feel free to unmute yourself or raise your hand. Yeah, Suzanne?
Yeah, so this is Suzanne. I was wondering, Scott, it seems as if everything went right from the advisory committee, and even the biostatistician at the FDA voted against it. But the FDA nevertheless ignored all of this, and they're supposed to hold everything to high standards. What else could have biostatisticians and the advisory committee could have done to prevent the approval that seems like a big mistake?
Yeah, so the answer is, by law, nothing, unless you have the ability to go in and charge individual FDA members with malfeasance, which is a tall order, and what you need to look at. The purpose of the advisory committee is to see what scientists believe. We don't all agree, and I'll say I have been on advisory committees where the majority went against, but close, and the FDA then approved it. I've been on ones, in fact, a month after this, I was on one where the committee was overwhelmingly say, change the indication only slightly, but the FDA went ahead and gave the broad expansion. And in retrospect, I say, yeah, do they have the right to change the terminology that's being used in the medical literature? So there's lots of things like that the FDA can come to us with one specific question, but that isn't the whole thing that they're doing. But in this case, this was the whole thing. I mean, they were asking our opinion on the whole thing, but they were very careful, I think, to try to avoid giving us opportunity to really tell them what they thought. If you want, you can go through and read the transcript. In particular, this was done with there was recording of the presentations beforehand and then brief presentations during the meeting. But Dr. Dunn stood up there and basically said nothing except for his conclusions. He did not talk about what the results were. He just said, I think this goes. I think this goes. And obviously, we questioned that in a major way. And we have written papers in both New England Journal and JAMA on this, various members of the committee talking about this. And again, that prior to the advisory committee meeting, there were people writing because the data had been presented at meetings, and they were writing papers just saying this does not match up. I had never read them prior to the advisory committee meeting. I don't work in Alzheimer's.
Thanks. There's a question from Ellen. Oh, Ellen raised her hand.
Yeah. I'm going to leave my video off because I'm on Wi-Fi. So, I mean, I guess since I do work in the Alzheimer's world, I've been sort of following this. I don't do drug trial or anything like that, but I think an awful lot of us in this field, certainly the clinicians that I know, were very concerned about the whole process and sort of felt that there was probably, politics going on between the Alzheimer's International Association or whatever, because we had a lot of sort of congratulatory emails from these sort of private groups about this, approval of the drug finally and so on. I don't know, Scott, if you had any indication of any of that kind of stuff going on, because it just seemed like it just seemed so improbable that this should have been approved. I mean, with all the data that was coming out in bits and pieces, it was a mess.
So, I mean, I can speculate on motivations that are there. Yes,
yeah.
I am not impressed with the Alzheimer's Disease Association.
Mm-hmm.
And in particular, the vehemence with which they were saying, oh, no, we need to approve this.
Yeah.
And you might imagine that their fundraising goes up when they say, we got a new drug approved. Mm-hmm. So just while I look at the sponsors being motivated by this, you can also think of somewhat, I call it the reverse Stockholm syndrome, but you can think of the people who are at the FDA and they get tired of, they probably have family with Alzheimer's. I mean, it's a terrible thing.
It's a terrible disease.
It's a terrible thing to say, but, sometimes, if they would just be hit by a car instead of watching them slowly go down. I wouldn't have wanted my grandmother to suffer when we first started noticing it, but by the last year and stuff, she couldn't do anything, and that was just painful for her family and things like that. So you can imagine that some of them are truly motivated. But that having been said, what I'm most alarmed at is how they trusted bad analyses by the Office of Clinical Pharmacology and over what the biostatisticians were saying. They just did not address it. They didn't understand issues there. And they just went through. So I am aware of another very questionable decision that was made by this Office of Neurological Diseases. Similarly, when the committee voted against it, it was more narrow, but with bad things. So possibly I'm complaining about individuals in that division. I usually work with the cardiovascular disease group, and I've had really no problems with them over the years. So that's what I can say.
Okay.
But it's not, to my mind, a mistake that there are two congressional investigations and the Office of the Inspector General looking at what the FDA did here.
My clinical colleagues, were appalled.
Yeah.
And they're not statisticians, but they still, they could see enough of the data to say, there's just the evidence just isn't there.
Well,
and this drug has serious consequences. I mean, this is not, they're not minor side effects. We're talking about major side effects. And they said, there's other drugs that work as well as this drug in terms of the change in cognitive change with much lower side effects, like maybe diarrhea or something.
Yeah, no, this ARIA is potentially serious. They did manage it all right during the clinical trials so that there weren't any disasters, but that's costly to do. So again, you're building up a lot of this in a really big way. So, it's my job to do the intent to cheat analysis. That whenever you're looking at things, you think about the worst case. And I will say that I can speculate on some aspects that go on with why Biogen stopped the studies for futility. I just can't believe that they didn't look at their results in more detail before they accepted the PMC recommendation. But that is an intent to cheat analysis that says, yes, knowing what I know, I could have told them, here's the way to cheat. So
yeah,
thank you.
Ted raised. Ted, I think, raised.
Ed.
Thanks.
Hi.
Hey, Scott.
Well,
So, coming from some more on the sponsor side, you made comments like, I think the sponsors would like this. And I will tell you, as someone in that space, that I think the only type of sponsor that might conceivably like this approach is someone that's sitting on data that's thinking, maybe I can rehash it and submit it, see what happens. The rest of the sponsors are looking at this in terms of the goalposts have changed. How should we be preparing our data? What do we need to do? It's not really liked. And so I'm curious for you to opine on a little bit is more, and you started going down this with the last question with Ellen. To what extent do you think that this reaction is something that was specific to the group evaluating this particular data versus the danger that this might sort of set another, lower the bar in a bad way for what the FDA deems as acceptable science? Because that's something that I personally would hate to see happen.
So, while lots of people, vilify big pharma and stuff, I'm not one to do it. And in fact, there was just an article by somebody who was in the Moderna clinical trial and who was saying they're out after money, and they did. And I actually think that they did wonderful jobs in all of the Operation Warp Speed and those clinical trials. And I felt that that was misplaced. But with small biotech, certainly I have dealt with the fact that accelerated approval is being used in cancer so much that when they come to me with a trial and I go, what are you collecting on a real clinical endpoint? And really, can't you do a randomized trial? And can't you do this well? And their answer is, oh, we're just going for accelerated approval, and then we'll sort that out later. So yes, that all of these things do move the goalpost. I like to believe that it wouldn't affect cardiovascular, where, I think they've done such quality work over the years. But again, this is not the first thing that I've seen from Office of Neurological Disease.
Tom, Fleming?
And just to throw in an additional thing, I have, in my experience, run into quite questionable practices by NHLBI several times. So I'm not as quick to say that the pharmaceutical industry is all bad and NIH is wonderful. I don't think that's true. So, Tom, you had your hand raised.
Yeah, I'd kind of like to go to the end, but I don't know how much more time Artie's going to give us. This is incredible.
5 minutes, I think.
Okay, then I think I'll jump in. So
sorry, Tom. There's also a question by Amelia that she had posted on chat. I mean, if Scott wants to see it.
Oh, sure.
Sorry, I missed that.
No, go ahead and take Tom's question first. That's fine.
Okay, I'll go ahead. So if we look at the totality of experience, FDA's influence has been remarkably positive, where we have come to trust and believe that they will bring the kind of rigor that we statistically should expect. But that's not a rule. That doesn't always happen. The question was asked already, what could statisticians have done? I'm remarkably impressed by what the statisticians have done within FDA, what the advisory committee did, what Scott's done. I've already said to Scott, he's my hero. The statisticians here have stepped up and brought, in my view, proper reasoning. And in most instances, while it's a collaboration, there is a consensus. And in most instances, the right thing has happened. And here, I want to touch on 4 or 5 things. Scott, just amazing presentation. My sense is that, first of all, I will limit my comments to what's in the public domain. And some of the really key issues here, as Scott's gotten at, is, if you're doing careful research and you see that there is uncertainty about what the right dose is in ApoE carriers, there's certain things you can change during a trial. You can change your endpoints. You can change your eligibility. You can't change the regimen. And shouldn't 301 and 302 have continued, and 303 and 304 have been started with the higher dose in ApoE carriers? And if not, then Scott, as you're getting at here, a futility analysis has to be very worrisome. I wouldn't do a futility analysis at immuno-oncology agents, checkpoint inhibitors, because the effects differ over time. They're because time post-randomization. Here, as you're pointing out, you've introduced a change that you would hope would enhance efficacy. And if it does, and you've implemented during the course of the study, then you shouldn't expect that what was often assumed in a futility analysis, that the early results come from the same truth as the later results. Another point that you raised is the timing of the cut for the analysis. I might differ. I differ very little in what you're saying. I might differ to say December is okay for a meeting in March. What would have been seen if the cut had been in February? The analysis that was done here, as you point out, can yield p-values, point estimates, confidence intervals. Was there, however, can you interpret that there's a clear specification of what the right timing is or cut of that analysis in this once you've stopped for futility? Is the right cut the December cut or the following May cut, or a cut when you in fact do have complete 18 months follow-up on all enrolled people? And is that clearly specified? As you point out, the concept of taking out patients because they deteriorated rapidly, and that's going to make your results look better, and in any way that's real, how is that conceivably influential? And the effects, even in the better trial, are half the MCID. And ARIA-E is real, and ARIA-E is really substantial. And so it seems at the end of the day, there's no logic here under any definition that this would meet a full approval. So, okay, then you fall back to accelerated approval when the advisory committee is told that's not where it's going to go. And then the data, when you look at this in what has been seen for amyloid effects being reasonably likely to predict. And in fact, what you have if I were a Bayesian, I would say, as you point out, you have 26 negative trials. Even a single positive trial without a negative trial doesn't give you a very good predictive probability. And so all of these issues here are just stunning issues in this instance, stunning issues that this would translate into an approval. And so my question, can this be at least a positive come out of this? And that is, isn't it time for us to rethink accelerated approval? And there is some rethinking, as you know, there's been a lot of advisory committee discussions lately. You've given those data summaries. But I'm wondering why we allow pricing at full level under an accelerated approval. You've taken any incentives away from a sponsor. You're giving them 9 years to get the truth here. Shouldn't we have done something? Shouldn't we be learning from this and doing something by saying, if you're going to give an approval for accelerated approval, the pricing is going to be basically cost pricing and not profit pricing? Your thoughts?
So, I mean, so I just today got a draft of a paper that comes from a think tank on accelerated approval that was primarily people in the cardiovascular world, and that they are in fact making a lot of suggestions, and I'm a member of that group. But of course, given what I was talking about today, they irritated me in the second line of the thing saying, well, we didn't really discuss Aducanumab 2 years ago, and so we're not putting it in this paper, and I'm going to have to fight that battle. But the idea is, England, again, puts in these constraints to say, yes, let's get the profit motive out of just getting a conditional approval, and realize that, the lifespan of a drug under patent is not as long as some of these confirmatory clinical trials go in. So I am not a fan of the accelerated approval. I do not think there's a very good record in the oncology world. I strongly urge people to go look at the FDA paper and then look at the et al. And Kesselheim papers, to see how that they're touting it works is not really true. And often, we could again, on that graph of the positive phase 3 studies, oncology was the lowest. Now, that was perhaps before some of these checkpoint inhibitors were coming as much to the foreground and we're getting success there. But I do think accelerated approval has to be fixed. That's a systemic problem as opposed to an individual clinical trial problem. So I tried to address both here.
So Scott, would you and others in the statistics area here, I mentioned, are heroes? We're not done.
Well, I hope that we can do some of this. I'll remark that I do have 3 former students who are in the statistical group at FDA, but I have very carefully avoided, talking to them specifically about this stuff. So, but I did talk enough to know that the biostatistics group was not happy with the FDA's decision.
Okay, maybe one last question, and the one from Amelia. So Amelia, do you want to pose the question yourself, or?
Oh, Scott already answered in the chat. Thank you.
Oh, I'm sorry. But I did it just to you privately. You asked on the futility, was it based on the primary endpoint or was it based on the surrogate? And it was based on the primary endpoint. There are trials that I work on that they sometimes do decide futility on the surrogate, but that's one where, it nearly comes to the level of harm by the time how conservative they're being.
Okay, this was great. Thank you again, Scott.
Great. Well, thank you for the opportunity.
Thank you, everyone, for joining us today. I will see you next week. Bye, everyone.
Bye.